As we continue to learn about the nervous system, we need to be able to identify and describe the anatomical features of the spinal cord and the spinal nerves, and to understand how those structures contribute to homeostasis by providing quick and reflexive responses to many different stimuli. The spinal cord is a pathway for sensory input to the brain and motor output from the brain. So we'll look at those ideas and then examine a spinal reflex arc, how they work, and some examples of those. So the spinal cord is made up of about 100 million neurons and even more neuroglial cells. Remember, the spinal cord is a part of the central nervous system that extends from the brain. The spinal cord and its associated nerves contain neural circuits that control some of your rapid responses to environmental changes. For instance, if you pick up a hot pan on the stove, those muscles grasp and then relax, so you let go really, really quickly and you drop that hot object even before you're consciously aware that it was hot or that it hurt. This is an example of a spinal cord reflex, which is a quick and automatic response to certain kinds of stimuli that involve neurons only in the spinal cord. So spinal nerves and the spinal cord processing. Now, after all that happens, of course, the brain says, ow, that's hot. But the spinal reflexes allow us to protect the body by having a very quick action before that information is sent to the brain for processing. In addition to processing reflexes, the gray matter of the spinal cord is also a site for integration or summing of excitatory postsynaptic potentials and inhibitory postsynaptic potentials, which we talked about when we were looking at neurotransmitters. The white matter of the spinal cord contains motor and sensory tracts, which function sort of as a highway along which the sensory information travels to the brain and motor output travels from the brain out to skeletal muscle and other effectors. As we begin looking at the anatomy of the spinal cord, we'll begin by examining the external structure. The spinal cord begins as an extension of the brain stem at the medulla oblongata and then travels through the occipital bone through that large opening called the foramen magnum and terminates at about the level of L2. Remember, that's the second lumbar vertebrae. Now, this is in adults. In newborn infants, the spinal cord extends to about the third or fourth lumbar vertebrae. But during early childhood, the spinal cord and the vertebral column are going to grow as part of the overall body growth. Elongation of the spinal cord, though, stops at about age 4 or 5, but the vertebral column continues to grow. So the spinal cord doesn't extend the entire length of the adult vertebral column. In most adults, it's about 16 to 18 inches long. You can also see a couple of very conspicuous enlargements. So the superior enlargement that you can see right up here where the spinal cord is much thicker, that's known as the cervical enlargement. So nerves to and from the upper limbs come to and from that cervical enlargement. The inferior enlargement, you can see here, is called the lumbar enlargement, and nerves to and from the lower limbs arise from that lumbar enlargement. When we begin talking about spinal reflexes, we'll see that they are initiated and completed through the spinal cord. Remember, we mentioned this a while ago when we talked about how that reflexive action allows you to drop that hot pan before your brain realizes that. Also in this image, you can see the spinal nerves extending out to and from the spinal cord, and you can see areas where you have an interweaving of all of these spinal nerves. So here in the upper part of the spinal cord in the cervical and brachial area, and then lower on the spinal cord in the lumbar and sacral area, you can see where the nerves all interweave together. Each one of those places is called a plexus. So we're going to examine the cervical and brachial, lumbar, and sacral plexus as we continue discussing our spinal cord. The spinal cord is protected. Remember, the tissues of the central nervous system, the brain and the spinal cord, are very delicate, and when they're injured or damaged, they really don't do a great job of regenerating. So these tissues are very, very highly protected, first by a layer of bone. So remember, the brain is protected by the cranium. The spinal cord is protected by the vertebral column. So if you remember back when we looked at the different types of vertebrae, one thing they all had in common was that vertebral canal, and that was the large opening. So here you can see the body of a vertebrae, and here you can see the spinous process and the transverse processes, and here's that great big opening, which was the vertebral canal, and that's where the spinal cord and its connective tissues that cover it are located. So we have the bone protecting the spinal cord. We also have cerebrospinal fluid, which is a liquid that cushions and provides basically a shock absorber and allows the nervous tissue to be pretty much in a weightless environment. Also provides nutrients and other things that we'll look at as we examine the cerebrospinal fluid. But for now, we want to talk a little bit more about those connective tissue coverings that are known as the meninges. So the meninges are these connective tissue coverings, some of which we can see very easily here, that are going to surround and protect that spinal cord. And while we have this image, I want to go ahead and point out a couple of things that we'll talk about as we proceed, but this is a really good place to see some of this as we're talking about the meninges, or the connective tissue coverings. We'll identify three different layers. So the outermost layer is the toughest layer, and that is the dura mater. The second layer, the arachnoid mater, is attached to that dura mater. So in a transverse section of the spinal cord and the vertebrae and the meninges, of course, you're not going to see that because it's not in living tissue, so it sort of collapses on itself. But you can see the area of the space here. Okay, this space is going to be filled with cerebrospinal fluid. Okay, so here you've got the spinal cord. Here you've got the subarachnoid space, subarachnoid below the arachnoid mater. Remember, your arachnoid mater is going to be attached here to your dura mater. And then we have lining the spinal cord itself a very delicate layer known as the pia mater. Another thing that we'll talk about when we're looking at some of the procedures that can be done associated with this area will be the epidural space. So epidura, remember epi means upon and dura, so this is the area above the dura, so here is the epidural space. Those connective tissue layers that we call the meninges are made up, as I mentioned earlier, of three different layers. The dura mater, dura means tough, mater is mother, so the dura mater is the most superficial of the three layers. It's very thick and strong, composed of a lot of dense, irregular connective tissue. The dura mater forms a sac from the foramen magnum in the occipital bone to the second sacral vertebrae. So it forms a tough connective tissue sheath. The second layer is the arachnoid mater, arachnoid, spider-like. So this is the middle layer of the meninges. It's very thin and avascular, provides a covering comprised of cells and thin, loosely arranged collagen and elastic fibers. It's called the arachnoid mater because of its spider web arrangement of these really delicate collagen fibers and some of those elastic fibers that create something that looks very much like a spider web, or very webby in its appearance. So it's deep to the dura mater and continuous through the foramen magnum with the arachnoid mater of the brain. So between the dura mater and the arachnoid mater is a very thin subdural space that contains interstitial fluid. Now remember, a while ago we saw epidural, that was above the dura. The subdural space is going to be below the dura. And the pia mater, the third of the meninges, pia means delicate, so this is the delicate mother, is the innermost thin and transparent connective tissue layer that lies on the surface of the spinal cord and the brain. So within the pia mater there are a lot of blood vessels that supply oxygen and nutrients to the spinal cord. And between the arachnoid mater and the pia mater is the subarachnoid space, which contains a lot of cerebrospinal fluid. Therefore, it's an ideal space for a spinal tap. So let's talk a little bit about this. So if we're looking at the distal end of the spinal cord, so there's an area called the conus medullaris, which is where the spinal cord tapers down and terminates. Below the conus medullaris, we have a structure called the cauda equina. Cauda means tail, equina is horse. So if you look here in this area, someone once upon a time thought this looked like the tail of a horse. So this is where you have a collection of the roots of the nerves that extend down below the conus medullaris. Now also below all of this, you have a structure that you'll see in lab called the filum terminale, which is an extension of the pia mater that extends inferiorly and fuses with the arachnoid mater and the dura mater and anchors the spinal cord to the coccyx. So let's go back up here now and talk about that spinal tap that we mentioned earlier. So remember, the spinal cord is going to end about the level of L2. So a spinal tap or a lumbar puncture is typically performed below that area. So a local anesthetic can be given using a long hollow needle inserted into the subarachnoid space. Now you can do that, of course, if you need to withdraw cerebrospinal fluid for diagnostic purposes. You can introduce antibiotics, or you can introduce anesthetics, such as would be done during what we call an epidural procedure when a woman is in labor, and this, of course, is going to deaden those nerves. And as we saw earlier when we talked about neurotransmitters and receptors, some of these anesthetic drugs will be done to deliver something that's going to block those sodium channels so the cells can't depolarize. So, of course, that is going to limit the pain response. So during this procedure, a patient's typically going to lie on their side with the vertebral column curved. So when they flex that vertebral column, that increases the distance between the spinous processes of the vertebrae and allows for easier access into that subarachnoid space. Remember, the spinal cord ends at the second lumbar vertebrae, but the spinal meninges and the cerebrospinal fluid extend to the second sacral vertebrae. So between L2 and S2, lumbar 2 and sacral 2, the spinal meninges are present, but the spinal cord is absent. So this provides a safe place to go in. It's usually done in adults between L3 and L4, or L4 and L5, the lumbar vertebrae, because this area provides a safe access to get into that subarachnoid space without risking damaging the spinal cord. So how do you find this? Well, you draw a line across the highest point of the iliac crest. That is called the supracristal line, and it passes through the spinous process of the fourth lumbar vertebrae. So that's a useful landmark for administering these spinal taps.